the release of neurotransmitter onto the dendrites is going to create electrical and chemical signals. The electrical potentials and currents that we generate in the dendrites are going to funnel into the cell body. They'll sum and determine whether or not we fire another action potential like we did in the last lecture. So in this lecture we're kind of closing the loop on the axon to dendrite to cell body cycle of neural signaling. First we're going to discuss kind of the nature of ion channel opening and closing and how ion channels work, because ultimately the synapse is going to create those electrical currents by opening ion channels. So when we look at synaptic currents, what we're looking at is the sum of many many individual channel currents. So we'll talk about channels and synapses first. Then we're going to talk about the passive and active conductance of charge through dendrites. We'll talk about the branching in dendrites and how that can create little logic networks, such as an AND gate, where two or more synapses have to be active at the same time. So one and the other must be active. Or they can create NOT gates, where only one can be active to cause an effect on the cell body. So if we want to depolarize the cell body, we need this synapse to be active, but not this one. So with that, you're probably imagining an inhibitory synapse, and you'd be on the ball there. In the end, we'll talk about dendritic spines, because these are pretty nifty. These are really flexible parts of dendrites that can grow and shrink or even disappear to modify the strength of synapses. Because ultimately, synapses are where we store our information. This is where we learn, it's how we remember things, it's through changing the strength of synapses. And dendritic spines are a great spot for that, because they're very flexible, and you can eliminate a dendritic spine without destroying the rest of the dendrites. So let's start off by talking about ion channels and single channel currents. So the single channel currents are all going to be kind of governed by chance, so everything's going to be a probability, and whenever we release neurotransmitter onto the dendrites, we're going to shift the probability of opening and closing, such that the ion channel is more likely to be open. Now, that doesn't mean that when we don't have neurotransmitter present that ion channels don't open up. Of course they do. It's just there's a low probability of it. And when we sum together all of the single channel currents, we create the synaptic currents. First, let's appreciate that there can be two parts of any synaptic current. So what we're seeing over here is an early and a late component. That's the E and the L. And so in part A, they're holding the cell at 4 different potentials. So we got a little stimulation artifact. Don't worry about that. That's just the electrical stimulation that's used to elicit neurotransmitter release. Then there's a distinct 2-phase response. Both of them are inward currents. We got our early and our late components. Early is going to be driven by ionotropic receptors, while the late component is going to be through metabotropic receptors. Now, not the receptor themselves. Instead, the metabotropic receptor is going to have some effect on an ionotropic receptor. So it's going to have to open an ion channel at some point. So it's not really affecting an ionotropic receptor, but it is affecting, let me be a little more clear here, an ion channel. Because it's really only going to be channels that are going to create appreciable currents. Now notice how this current changes, or how the potential changes, as the cell becomes more hyperpolarized. So when it's a little depolarized at minus 66, we get an inward current here. If we drop it down around minus 76 millivolts or so, we get our stim artifact, and then basically nothing at that early phase, and all we get is the late current. It's a little smaller, of course. This would be the reversal potential, because I want you to notice what happens when we go even more hyperpolarized here. So now we're going below rest, we've got our stim artifact, Now we actually have an outward current for the early phase. So you'll notice right here it reverses. And that's why we call them reversal potentials. When you're above that, so when you're depolarized relative to it, we're getting an inward current. Whenever we go hyperpolarized, our early current is now outward. And then they're plotting this in part B. This is not really what you'll see. You'll see something very similar to this called an IV plot, where we'll have current on the Y at different membrane potentials. That's why it's an IV plot. So you look at how changing the membrane potential affects the currents that you get. Now this is made up. These are just simulations here in A, because if you're holding the cell at a potential, you're not going to measure potentials because you're in, well, voltage clamp. So you'd have to be measuring currents. Anyway, that aside, what you'll notice here is that we have two distinct reversal potentials. So at minus 66, we'll put that here, we had an inward current. At 76, nothing, and at minus 102, which I guess would be over here, we have an outward current. The point at which we had no current, that's of course the reversal. So that should tell you that the early current is brought on by chloride. And that late current that disappears around minus 102, that's going to be brought on by a potassium channel. So this is most likely a GABAergic synapse. They dumped out GABA, stimulated GABA A, which is a chloride channel for the early current, and they stimulated GABA B receptors, which are metabotropic. And most likely you're going to get the beta-gamma complex interacting with some sort of G-protein stimulated potassium channel to create that late current mediated by potassium. So when we're looking at synaptic currents, we can have early phases, we can have late phases, and they can be mediated by different ions. It depends on what receptors you have there. What we're going to focus on right now is just the ionotropic receptors. Just the ion channels that open up whenever we dump neurotransmitter onto them. Now we can record the activity of individual channels with single channel recordings. And the way that we do these is to first carry out patch clamp recording as you would normally. So let's say this is our cell. You'll lower your pipette down onto the cell. Now if you pull your pipette away, the cell is most likely going to stay in place. Sometimes you rip the whole cell out. But the cell is most likely going to stay in place, and what will come with you whenever you pull your pipette away is a little patch of the membrane that was attached. Now hopefully you have yourself an ion channel in there. And what you have is the inner portion of the receptor exposed now to the solution because you just pulled it back. And you just plucked off that little bit of membrane. So the outside of the cell is inside your patch pipette. We call this an inside out because the inside is pointed out. If you want to get the outside out, what you can do is first go whole cell. So apply a little negative pressure, get rid of that membrane, and then when you pull your pipette off, the kind of outlying areas of the membrane are going to fold back. Because when the hydrophobic core of the membrane is exposed, it's thermodynamically unstable. So it rapidly folds back to form one continuous sheet. So in that case, when you pull your pipette off after you've gone whole cell rather than cell attached, that little bit of membrane that you pull off with it is going to fold back in, and you now have an outside out patch. So when that flops back, now the outside is going to be pointed outward. The ion channels that are in there, you can then record their activity. So you can apply neurotransmitter onto them. So this cartoon is showing you that. When they spit out, they use a little puffer pipette to apply a little bit of acetylcholine. And when you do that, you're going to see these square waves. And that's because ion channels are either going to be open or they're not. And when they're open, ions move. So when the ion channel opens, you see an inward current. And when it closes, it goes away. It's a square wave here. We're dealing with essentially no membrane, so we don't have much capacitance. So we actually are going to see square waves in this case, no capacitance current. Or at least so little that you won't really see it. Now I want you to notice is that this is opening and closing several times, even though the entire time we're delivering our acetylcholine. So the ligand is present the whole recording. But you see multiple opening and closing events. And that's because the opening and closing of ion channels is probabilistic. It's not that neurotransmitter binds and it opens the entire time that the neurotransmitter is there. It might not even open. And it's also the case with voltage-gated ion channels that once you cross threshold, you might open, you might not. You might open for a very long period of time, you might open and then quickly close, you might open and then quickly inactivate. So it's chance. So here's some actual recordings. These are from voltage-gated calcium channels. And what I want you to notice here, it's still digital. It's a little noisier because it's the real world. And it's still probabilistic. Rather than delivering a little bit of neurotransmitter, what they're doing on top there is delivering a potential change. So they're going to hold that little patch of membrane at a different potential. That's what then opens up voltage-gated calcium channels, and then they get their currents. And I want you to look at these, compare part A to part B. So the top, that's with nothing special going on in the bath, and then B, same thing, but they're adding a little ATP. And what ATP is going to do is bind to the extracellular face of voltage-gated calcium channels and increase their open probability. Now I want you to notice the entire duration of that square wave. If you do a few different recordings, we got 1, 2, 3, 4, 5, 6 recordings. They're not all the same. That same ion channel didn't respond the same to the same stimulus. On top, it opened up and then closed. In the next one, didn't do shit. In the one below that, it opens up for a whole lot longer. So you'll notice you don't always get the same response from the same input. And that's because ion channels work based on probability. So their opening and closing is probabilistic. And we call it digital because it's either open or it isn't. You get a reliable current. When it opens, you get the same amount of ion flux. If the channel's open, ions move. If it's closed, they don't. And if it's blocked, they don't either. So whenever we talk about desensitization of ion channels, what we're doing is just decreasing open probability. And open probability is exactly what it sounds like. If we were to look at this period of time where it should be open, how much time is it spending open? We'll call this all the time, so we'll call it 100% of this. It's open 0% of it. In this case, we'll say it's open, 30% of it. So then you would just take your average of these. So we'd say that the open probability in this case, whatever this is, pretty close to 30%. Is that right? Probably not. Anyway, whatever this is. Not 1/3. I don't want to do the math on that one. Okay, 43%. Much better. So we call this one a 43% open probability. It's spent 43% of the time open, and about 57% of the time closed in these recordings. When a receptor desensitizes, this value is going to drop. So in a desensitized receptor, what we'd see is, now these might close up a little more often. Maybe here we got a couple of closing events, can reopen. But it's spending less time open whenever we see desensitization. This amplitude, though, does not change. That's the key feature. It's always going to give you that reliable current, so long as you have the same driving force. You know you're going to have the same ion imbalance. That's constant. But at a given membrane potential, you should have a reliable current from each individual channel. And you can, of course, calculate that with Ohm's law. It's written right there for you. So the single channel currents, I, is going to be equal to Vg. G in this case is going to be the conductance, so the inverse of resistance. Different channels will have different conductances, but for each given channel it'll have a reliable conductance. And then how far are you from that ion's reversal? So if it's a voltage-gated calcium channel, if you bump it all the way up to 125 millivolts, you won't see a current. You're already at reversal. So V is driving force anytime we're trying to calculate currents. The further you are from reversal, the greater the driving force, the greater the current. The closer you are to reversal, lower the driving force, lower the current. When we think about synaptic currents, what we're doing is summing together all those individual currents. So part A is showing you the individual currents. Let's say we consider about 5 different receptors. Some of them are going to open for essentially no time. Some are going to open for a moderate amount of time. Some are going to open for a longer period of time. Most likely, you're going to see the highest probability of opening near the beginning of the delivery of your neurotransmitter as it diffuses away, as it unbinds from the receptors. That's, of course, a chance event. As the receptors just spontaneously close, these currents are going to terminate. So when you sum together all of those individual square waves here, what that gives you is the smooth synaptic currents that we saw earlier. In this case, we'll just consider one phase. We just have that early phase here. But this, that smooth movement down and up, part of that's going to be because of membrane capacitance. And the other part of it is going to be because of the summation of those individual ion channels. So there's actually a whole bunch of square waves that are creating this, but they're so small we don't see them. Each individual ion channel is going to create picoamp currents, but we're going to be able to measure much larger than that. Well, hundreds of picoamps or nanoamps of current, depending on the synapse and your access to the cell. So we're still looking at V equals IR down below, but whenever we're thinking of the synapse, what we have to do is just sum all of our individual single channel currents. So we still have driving force. So how far are we from reversal? We still have our single channel conductance, but now what we're going to do is multiply that by how many ionotropic receptors do you have? So how many channels are there? If you pack more channels into a synapse, you're going to expect a stronger synaptic current. And then the open probability is also going to factor in here. If your receptors have a high open probability, you'll expect a larger current. If they have a lower open probability, you'll expect more of them to be closed, and thus you'll have a smaller current. It's still just V equals IR. We're just essentially taking an average, or I guess a sum, of our individual single channel currents. But it's still V equals IR. And so is this. This is taking into account leak. So we can't forget lecture 2. All right, the resting membrane potential still exists, and it's still established by ion channels as well. So the leak current that we're generating at rest is going to tend to hold us near rest. That's all this is saying. So if you look on top, the leak current is going to equal your leak conductance, of course, times the driving force. So how far are we from rest? So your leak current is going to be fairly low at rest because you'll already be at reversal. In fact, if we are at rest, we should have no leak current. As we move away from rest, then we start to create our leak current. It's still V equals IR. Now what we have to keep in mind is that that leak current is always there. So whenever we open up our synaptic currents, that leak current is going to offset the synaptic currents. So we have to correct our expected potentials based on our leak current. And the big idea here is that resting membrane potential is going to act to inhibit changes from resting membrane potential. It's just that simple. Cells that are resting at -70 want to pretty much stay at -70. They're going to resist movements away from that. So you're going to need stronger currents in that case. And the leakier the cell, the stronger the depolarization you need. So different cells are going to have different degrees of leakiness. And let's take a look at the math here to see how the leakiness of a cell is going to impact its excitability. So whenever we're calculating leak-corrected potentials, we're going to look at, of course, the potential change that we get from the synaptic currents, and we're going to take into account the leak current as well. So the formula is given here. You can always look this up. Don't worry about remembering it. You should just understand from a very logical standpoint that leak current is going to hold you near rest. The more leak current you have, the harder it is to move away from rest. So let's look at this. So at rest, we have no synaptic input. We're going to say we're resting at -70, and what I've done is give kind of a generous, but nowhere near out of physiological ranges here, 10 nanosiemens of leak current. So we're at -70. Whenever we get some synaptic input, so we're going to open up some channels. These are going to be non-specific cation channels, so they reverse at 0 millivolts. And we're just going to have a 0.2 nanosiemens conductance. So it doesn't really do anything to the numerator, but it does affect the denominator. So we're not going to be at -70 anymore. We're going to be at -69 millivolts. We got a whole 1 millivolt of depolarization because the cell was kind of leaky. Now if we tighten it up and we only have 1 nanosiemens of leak current, that same synaptic input is going to cause 12 millivolts of depolarization. So the leakier the cell, the harder it is to move away from rest. So leakiness is going to equate to a lack of excitability. And that should make good intuitive sense. Now the types of receptors that we have are going to be either excitatory or inhibitory. We're either going to move toward threshold potential, and ideally well above it, or we're going to move away from threshold potential, or we will resist changes, more likely. Now this is all about ionotropic receptors. So you really need to only think of about 2 things. For excitatory, just think about non-specific cation channels. Most excitatory ionotropic receptors are going to be non-specific cation channels, or there'll be something very close to that. There's about the same amount of positive charge in the cell, in this case it'd be potassium, as there is positive charge outside. In this case, sodium, calcium, lithium, magnesium. So all the positive charge, whether it's monovalent, divalent, doesn't matter. If you're a non-specific cation channel, you let any old positive charge move. The only way that you're never going to get a net movement of positive charge, given that it's basically equally concentrated, is when you have 0 charge at the membrane. Anytime the cell's a little negative, positive charge is going to want to move in. Anytime the cell's a little bit positive charge is going to want to move out. So the reversal potential for a non-specific cation channel is going to be 0 millivolts. This doesn't mean that we create no current. It means that we move towards 0, and that's well above threshold. The purpose of excitatory currents, or excitatory potentials, is to cause the neuron to fire an action potential. We don't actually give a shit about sub-threshold changes in membrane potential. They don't affect whether or not the neuron communicates with its target. All we care about is the neuron firing an action potential or is it not? So the purpose of excitation is to increase the likelihood of firing an action potential. And we do that through depolarization, of course. Now, for inhibitory postsynaptic potentials or currents, we need to think about chloride channels. That's what it's all about. It could be potassium, but it's not. And you know that it's not because I just told you it isn't. That late current that we get can definitely be potassium. So the late current is going to be through metabotropic receptors, and in that case you can definitely see potassium currents. But that's not going to be through ionotropic receptors. We're going to have different potassium channels for that. For fast inhibitory synaptic currents, it's all about chloride, and these of course reverse basically at rest. So we're not really going to cause a whole hell of a lot of hyperpolarization because we basically have no driving force. Instead, what we're going to do is prevent changes in membrane potential. We're going to prevent reaching threshold. We don't really care about hyperpolarizing a resting neuron because it's already not communicating. What we're trying to do is to prevent it from communicating in the future. So that chloride conductance is going to open up, but we get basically no current until we start to depolarize. So as we move away from chloride's reversal, then we get an increase in driving force. Then we get a chloride current. And this is what we call shunting inhibition. Because that positive charge that comes in from electrical input essentially gets removed. That's what we're thinking of it as. We're shunting that positive charge through inhibition. So take a look at the cartoon here. All right, we got basically, it's a monovalent cation channel on top. But most are going to be nonspecific. And then we got a chloride channel on the right, so excitatory, inhibitory. If you look at the effect of excitatory input without inhibition and with inhibition, you can see that inhibitory stimulus there decreases the window of excitation. But really what it's going to do, because of that, is make it much harder for the neuron to fire an action potential. You're going to need stronger excitatory input to finally spike. So the top is showing you the same thing that we just saw. Basically, you have your excitatory input without, that's the dark line there, and then with inhibitory input. Notice smaller degrees of depolarization. Thus, you need stronger depolarization to hit threshold. The bottom is showing you the FI plot, essentially. So the frequency of action potential firing on the Y, and then the amount of excitatory input on the X. You need a whole lot more excitatory input to start to see an increase in firing. That's what inhibition is all about. Now, for our synaptic currents, we're summing up many individual single channel currents, but in the cell body, we're summing numerous synaptic currents. So, the summation of synaptic currents is not going to always work out in a linear fashion. That is, 1 plus 1 isn't always 2. In fact, it's almost never 2. So we can get what we call sublinear summation because of receptor desensitization or a change in driving force. So this will be our expected potential, and this will be our measured potential. If it's linear, these should match up. So if we expect 1, we get 1. We expect 2, 3, 4, we get 2, 3, 4. So for linear summation, let's say that we have input at 2 different synapses. Here's a dendrite. Let's give it a cell body over here. If we were to just stimulate synapse 1 or synapse 2 individually, let's say that each of them gives us 1 millivolt. So I stimulate synapse 1, I get 1 millivolt of depolarization. I stimulate synapse 2, I get 1 millivolt of depolarization that I'm measuring over here in the cell body. So if I stimulate both of them together, I should get 2, right? Of course not. That's not going to happen. Because as I stimulate these, that charge, of course, spreads in all directions. And that depolarization is going to affect the driving force. So in a synapse that's a little depolarized, we're going to have decreased driving force for our excitatory currents. And as such, they won't sum in a linear fashion. So that illustration over here is showing you that. The plots on the bottom are showing you 3 different synapses, and then synapse 1 and 2, what you'd expect arithmetically, that's the gray line there, and then what do you see whenever you actually measure it, that's the black line. So 1 and 1 should give us 2, but it doesn't. It gives us something less than 2, and that's why we call this over here sublinear. I guess I can write that. Sublinear summation. We're getting less than we would expect, but that's not always the case. Sometimes we get supralinear summation. We actually see more than we would expect. And when you see that, you should be thinking NMDA receptors. So NMDA receptors, I'm sure you're all well aware, are blocked by magnesium. So we got a magnesium ion actually sitting down in the pore. Only whenever we have a little bit of depolarization already, so we have a buildup of positive charge, does that repel magnesium and unblock this channel. So NMDA receptors are both voltage gated and ligand gated. And we'll see this in a little bit later in this talk. But whenever that depolarization spreads over here, what it does is unmask additional ion channels. Because you now have a greater number of ion channels that can move ions, you're going to get a greater current and a greater potential change. So it could be sublinear, it could be supralinear. It's usually not dead-on linear. Sublinear is caused by desensitization. So after ion channels conduct ions, they then decrease their open probability. This is a way of preventing the neuron from being overexcited. It's a way of making sure that signals are relatively brief. Different receptors desensitize more or less, but AMPA receptors, which are your kind of run-of-the-mill, basic, fast, excitatory transmitters, desensitize rapidly. So you're going to see desensitization. So if you stimulate a receptor over and over and over, you're not going to get as strong of a response. And also because of a change in driving force. Supralinear, NMDA receptors. Okay, now I can sleep. So once we bring in charge then, whether we get the amount that we expect, a little less or a little more, it's going to then move through the dendrites. It's going to move passively, and if it encounters some voltage-gated ion channels, there's going to also be active propagation of charge. So dendrites are not just passive cables, but we're going to think about them like that for just a second. So of course the movement of charge is going to be based on the time constant as well as the space constant. So we got to think about capacitance, we got to think about resistance. That's going to affect how charge moves. I'm not going to hang out on this point because we've already covered it. Now what we need to keep in mind is that although dendrites are cables, they are not cables of uniform length. Of uniform diameter. Dendrites taper. So as you move further away from the cell body, this thing actually decreases its diameter, and thus it changes its internal resistance. Much higher internal resistance here because of the smaller diameter in the distal dendrites than we see in the proximal dendrites. Still fairly high internal resistance, but less here than here. And that explains what we're seeing in this figure. So I want you to look down in panel C there. That's showing you triple patch clamp recordings in distal dendrites, that's the green one, proximal dendrites, that's the blue one, and the soma, that's the red one. And I want you to look at the amplitude of the response in distal dendrites compared to proximal dendrites compared to the soma. Distal dendrites with higher resistance are of course going to have greater potentials. Why? You already know the answer. I know you do. It's Ohm's law, of course. V equals IR. Increase resistance, increase the potential for the same given current. Now, of course, changing that resistance affects how the current moves through the dendrites. Your space constant is going to change. So the passive propagation isn't going to work as well whenever we have areas of higher internal resistance. This will kind of bias charge towards moving to the cell body if it's completely passive. And we're also going to have a change in capacitance too. So as we have wider dendrites, there's more membrane than in thinner dendrites. As such, the time constant is going to change a little bit. So you're going to see taller and briefer spikes out here in the distal dendrites compared to the more proximal dendrites. So they'll spread out a little bit more because of a subtle change in tau, because of the change in capacitance. What we also need to keep in mind is that we have branching of our dendrites. It's not one smooth cable. There's a bunch of cables coming off, and at every branch point, we're going to cut that charge in half. So the charge that's moving here is going to go that way and that way. It doesn't know which way to go. So at every branch point, we're going to be splitting our charge in half. Now, with the passive properties, you're going to see exactly what this is showing you right over here in panel C. Even though you have measurable changes in potential in the distal dendrites, basically nothing is going on in the cell body. And when you look at that plot on the bottom right, as you move away from the cell body, so as you move further on the right in the x-axis, notice the red dots, they're decreasing. So the somatic potentials are dropping when synapses are further away from the cell body, even though those synapses have greater potential changes. So if dendrites are just passive, distal dendrites are meaningless to the neuron. They have no impact on somatic potentials. So they don't need to exist. They're a complete waste of our time. Of course, that's not always true. Dendrites aren't always going to be passive. We can have enough depolarization to stimulate active conductances. Dendrites are excitable. You don't always see that drop in somatic potential as you move away from the cell body. So here's another set of data showing you how active conductances in dendrites are actually going to essentially normalize somatic potentials as a function of distance. So it doesn't matter if you're 300 microns away from the soma or 100 microns from the soma. Those dendritic potentials will be much larger when you're 300 microns away, but by the time they make it to the soma, now we're getting a fairly consistent 0.2 millivolt depolarization, regardless of distance. So look at E here. That's showing us that most clearly. D is showing you exactly what you'd expect. Distal dendrites have much greater potentials because they have higher resistance. But the charge that makes it to the soma is fairly consistent. And that's because dendrites aren't just passive cables. They are excitable too. So when the neuron fires an action potential, it only moves down the axon. That's it. Because that charge knows it doesn't need to go anywhere else. I don't need to go up into the dendrites. Those are just the ears. I need to head down the axon. This is where I'm going to cause the release of neurotransmitter. So action potentials only move down the axon. In cartoons anyway, but in the real world, charge doesn't know where to go. Action potentials are going to back propagate into the cell body and into the dendrites. Because charge doesn't have a mind, it just moves randomly. So we're going to get back propagation of action potentials. And in fact, action potentials can back propagate into dendrites and stimulate action potentials within the dendrites. Now the dendrites are of course less excitable than axons. They're not axons, right? They do have some voltage-gated ion channels, but they also have ligand-gated ion channels. Because their primary job is to listen, and they can only listen to the chemical transmitters that are coming in through ligand-gated ion channels. They have a bunch of other ion channels too, so they still have voltage-gated sodium channels, calcium channels, potassium channels, but they're just they just don't have quite the density as we see in the axon. So they're not quite as excitable, but they're still excitable. Now, how excitable they are depends on what ion channels they contain, and also, of course, the degree of branching. So take a look at this illustration here. Again, triple patch clamp recording on top. We're in the axon, that's green. The cell body is blue, and the dendrites, orange. They stimulate an action potential. We see it in the axon. Of course, it back propagates to the soma. That's how we record them in the first place. And then it makes it out there into the dendrites too. It takes a little bit, but it makes it. A little less on the amplitude, but definitely a detectable spike. So dendrites are excitable. They're not just passive cables. Now, how well that action potential back propagates depends on how much branching there is. Take a look at that Purkinje cell there. Tons of branching, and at every branch point we're splitting the charge in half. So our ability to then stimulate the next round of active conductances drops. And so you'll notice the Purkinje neuron has a fairly rapid drop in the amplitude as you move away from the soma. So this is distance, and this is going to be the amplitude of that back propagating action potential. So our Purkinje neuron is going to drop dramatically because of the branching. But in pyramidal neurons, for example, as we move up that apical dendrite, it's not as pronounced, and that's because there's just fewer branch points. Now, the ion channels that are in dendrites are a little unique to that in the axon. So we're going to see a little lower levels of sodium channels, but we're going to see more of the inactivating potassium channels, and we're going to see some HCN channels as well. So we have a variety of voltage-gated ion channels. That's the point here, and they're going to do different things to the dendrites. So, of course, the potassium channels are going to cause some hyperpolarization. That's going to hold our dendrites at a little more hyperpolarized potentials, and that'll, of course, increase the driving force for excitatory currents. The HCN channels, those are going to cause depolarization, but actually in doing so, they're going to inactivate or desensitize some of the voltage-gated calcium channels that are present, and they'll actually cause hyperpolarization, believe it or not. Let's look at the data and then we'll see if we believe it. So we're looking at calcium imaging in the dendrites on top, and we're looking at somatic excitatory postsynaptic potentials on the bottom. Black is a control mouse, and red would be an HCN knockout mouse, so no HCN channels in their dendrites. On top, you can see that calcium current that we get is much longer lived in our HCN knockouts. And that's because we haven't inactivated some of the voltage-gated calcium channels. So they're able to be opened up and conduct calcium. You'll notice in the bottom that the cell is actually a little more hyperpolarized in the HCN knockout. That's because of a lack of that depolarization from the HCN channels. And we're going to see more prolonged excitatory postsynaptic potentials because we're not inactivating some of those active conductances in the dendrites. So we have a variety of ion channels. That's all it is. You should be able to look at this and make sense of what's going on. It makes sense that it's hyperpolarized because we've removed depolarizing currents. And it makes sense that the postsynaptic potentials last a lot longer because we haven't inactivated as many of the active conductances in the dendrites. Now, dendrites are able to initiate action potentials. So it's not just the case that action potentials have to back propagate. We can actually stimulate action potentials out here in the dendrites. And we call these a dendritic spike. It just requires very strong input. So, two different recordings here. On top, it's with weak stimulation in the dendrites. It's enough to bring the cell to threshold And so you'll see in the soma, it fires an action potential, and after that we see a spike in the dendrites. So when they superimpose those on the right, first soma, then dendrite action potential. On bottom, when you have a very strong input to the dendrites, you of course get an earlier action potential in the soma compared to the one above. But what you're getting first is an action potential in the dendrites. So have a look when they superimpose those. First dendrite, then soma. So with very strong input, we can sufficiently activate the active conductances in the dendrites and create a dendritic spike that's going to travel down and create much stronger depolarization here in the cell body. So with weak input, we get somatic action potential that backpropagates. With very strong input, we get a dendritic action potential that forward propagates to cause more immediate firing of action potentials in that neuron. So we can initiate action potentials in the dendrites. They're not just a function of action potentials generated here backpropagating. Now the spikes that we see in the dendrites can be mediated by calcium, sodium, or both. So the calcium spikes that we see in dendrites, these are unique. You don't see calcium spikes in axons. You see sodium spikes. Now we also see those in dendrites, of course mediated by voltage-gated sodium channels. But in the dendrites you can see pure calcium spikes, where voltage-gated calcium channels open up. These last a little bit longer, so you'll notice the dendritic spikes here, if you compare those to the spikes in the cell body, they're a little bit wider. And that's because of the use of calcium. So these calcium spikes are typically going to be caused by backpropagating action potentials that then open up voltage-gated calcium channels. So if you look at the recording on the top, what they're doing is stimulating action potentials in the cell body, and they're measuring the membrane potential in the dendrites. So what they then do is record it in the presence of tetrodotoxin. So they deliver very strong depolarizing currents into the cell body, and when those backpropagate, you can see a spike. Now you shouldn't see a spike if this is mediated by voltage-gated sodium channels, because tetrodotoxin blocks those. So in the presence of TTX, we still get dendritic spikes when they're mediated by calcium. We don't see them when they're mediated by sodium. That's down below. That's a sodium spike. These are a little faster, and that's because of the activation then inactivation of voltage-gated sodium channels. So they're a little bit faster. And these are going to be driven more by the excitatory input out there in the dendrites. That's more typically going to cause a sodium spike. So when we get strong excitatory input, and that's usually going to require input at multiple sites near one another all at the same time. But when we get a bunch of excitatory input, we fire a sodium spike. And we know that the one on the bottom is a sodium spike because we added TTX. And when we add TTX, that initial spike is lost. All we're left with is the very strong depolarization, but you're not noticing that spike. In a very special case of dendritic spikes would be NMDA spikes. These are also, of course, unique to dendrites. That's where we find our NMDA receptors. And these are initiated by glutamatergic input. But of course, NMDA receptors are blocked by magnesium. So in order for this to work, we have to already be depolarized. And in order for these to propagate, we have to continue to get glutamatergic input. So this is a way for neurons to create those AND gates. We're going to look at this later, but we'll go ahead and bring it up now. If we receive strong input here, and we initiate an NMDA spike, that's going to move into the dendrites. Of course, it's going to get cut in half at every branch point, so if we want to propagate this, we're going to need additional input along the way. And if we get appropriately timed input, so first we get our spike, and we continue it along, that spike is going to make it all the way to the cell body and cause very strong depolarization. So here we have multiple AND gates. If these and these and these and these synapses are active, then the NMDA spike makes it down to the cell body and we get a much stronger response than we would get if they weren't all active. So the tapering of dendrites and that branching is going to affect how charge moves through, and it's going to affect how charge interacts with each other. So because of this branching and the loss of charge, we're going to need refreshers if we're going to carry that NMDA spike forward, for example. And that creates the AND gate. So here's just a little simplified example I have for you. So in both cases we're looking at cats. Now the one on top is a nice cute little kitty cat. And I'm going to tell it's a cat, and since it is a cat, I'm going to get input from the cat synapse. I have another synapse along the way that's going to tell me, how frightening is this? Should I be worried, in other words? And I'm not. I look at that cat, and I'm happy. It's kind of cute, and its head is cocked to the side, so it's even cuter. Now, the little kitty cat below is not a cute little kitten. It's a tiger, and it's showing its teeth. It looks kind of angry. I see that it's a cat, so my cat synapse is active. But I also see that I should probably be afraid of this. So the excitation that I've created in that distal synapse is going to get carried forward by the input from that, what I'm calling a salience neuron, that says, you should worry. So now my panic neuron gets a much stronger response. Am I going to panic from looking at this cat? Yes. So that's the AND gate. Only when they're both active is this neuron strongly excited. And that's because those distal synapses, they're going to be losing their charge at all those branch points, so we need a refresher. So those NMDA spikes make a whole lot of sense for mediating the AND gate, because NMDA receptors are essentially an AND gate. We need two things to happen. We need the neuron to be depolarized, and we need to have glutamate and glycine present. So what we're looking at here is a real IV plot for two types of glutamate receptors, AMPA and NMDA. AMPA receptors are your basic run-of-the-mill excitatory neurotransmitter receptor. If you dump glutamate onto them, you'll get a current. And these are both pretty much non-specific cation channels. It changes a little bit with AMPA receptors. But at 0 millivolts, we get no current. When you're negative, you get an inward current. When you're positive, you get an outward current. Not the same with NMDA receptors. There we typically see a J-shaped curve. And that's because at negative potentials, we have the magnesium block. So only when we're depolarized a bit and we have neurotransmitter bound do NMDA receptors move ions. And when they move ions, they move a whole hell of a lot more ions than AMPA receptors. NMDA receptors are going to, just as a population, be more permeable to calcium. And calcium is an important secondary messenger that's going to stimulate kinases and affect how the synapse works. And we think this is really important for memory. And I'm going to talk to you more about that in Lecture 10. But NMDA receptors also don't desensitize as rapidly as AMPA receptors. And so you get more prolonged synaptic currents. So those NMDA spikes are going to be propagated throughout the dendrites at sites where we have input. So that's why we're going to get that supralinear summation. And that's what this is showing us. So there's stimulation of a couple different types of fibers in this experiment in order to generate an NMDA spike. So when we pair stimulation of thalamocortical and corticocortical inputs, so we're getting coincident input onto this cortical neuron. So the neuron they're showing you there lives in the cortex, and it's going to have input from the thalamus and the cortex. So they can stimulate both sets of fibers to get coincident activation. And that's what you need to stimulate NMDA receptors. And when you do that, one and one, is greater than 2. So if you look at the traces on top, this is just showing you the voltage in the cell body whenever you're stimulating just thalamocortical fibers, that's green, cortical fibers, oh, that one's green. Thalamocortical is kind of a cyan. Sorry, it's hard for me to see here. The dark blue is what you'd expect if they just summed, like numbers do, if 1 and 1 truly equaled 2. So if you just took their two traces and summed them, you'd get the blue. What you really get is what you see in the red. When you stimulate both at the same time, you get a much stronger response. And that's actually because of NMDA spikes, and they've recorded those below. So you can see in a few different areas of the dendrites there, they found 5 of them. They're only showing us 2, the red and the blue. But the yellow and the orange and the green, those also had spikes. Might be yellow, might be chartreuse. It's hard to say. Anyway, the colored ones, not the white ones, they saw NMDA spikes kind of propagate along the dendrites, and they see hot spots because it's at the areas, damn it, it's gone. It's at the areas where we got that input. There we're seeing the hot spots that are keeping that NMDA spike going. Because we're only going to stimulate NMDA receptors when we have depolarization and glutamatergic input. So the NMDA spike can essentially disappear between these branch points when you're just looking at calcium, because you won't get calcium influx without opening NMDA receptors. The charge will spread, but the calcium won't. You'll just see calcium at the sites where you're releasing neurotransmitter. And what that's doing is refreshing the NMDA spike, and that's why 1 + 1 is greater than 2 in the top. Now if they add AP5, that's the gray trace, it basically looks like the blue one. It's a little bit lower at the beginning, but it's, it's close. 1 + 1 is basically 2. It's 1.8 in this case. But we don't see that supralinear summation because we've blocked our NMDA receptors with AP5. And the reason that we're going to get supralinear summation in the cell body is by carrying forward NMDA spikes. These things are nifty. So that's our AND gate. As long as we get input here and here and here and here, that neuron is going to respond much stronger than it would if we just had one of these. So they have to happen together. It's an AND gate. We can create NOT gates as well with inhibition. And that inhibition is much stronger when it's on path. In other words, between the excitatory synapse and the cell body. So let's consider our on and off path inhibition. So we'll have our neuron and our excitatory synapse be black here. So we get, let's say, excitatory input here. This will be releasing glutamate. Let's get ourselves a purple GABAergic neuron, and let's put our inhibitory synapse right here. This is going to have a more pronounced effect on the currents here than if I had a synapse closer here, but not between the synapse and the cell body. In other words, having it off path. And it'd be even weaker if I were off path and on a different branch. And all of that is seen here. So if you look on the top, all that's showing you is that on-path inhibition has a dramatic reduction in that excitatory postsynaptic potential in the cell body. That's what they're plotting. So that positive deflection in membrane potential becomes a whole lot lower whenever you have inhibition. And that's because of shunting inhibition. So as that positive charge flows toward the cell body, that's going to increase the driving force here, and we're going to get a much stronger chloride current as a result. Much stronger inhibition compared to off path. So if we were to stimulate here, we'd expect very little effect. Some, but very little. So this would be a NOT gate. If we want this neuron to be excited, we need this, not this, to be active. And the last thing that we need to talk about are dendritic spines. So right now we've just kind of thought about input onto dendritic branches, but some dendrites have little spiny protrusions called dendritic spines that are going to make stronger and more flexible synapses. Now almost always these are going to be excitatory, but they're not always. Notice I said almost always. So in adults, most of the time you have a single excitatory synapse at dendritic spines. In some cases they look to be inhibitory because they are symmetrical in an electron micrograph. So there's probably inhibitory synapses on spines as well. In some cases they split and form two synapses. So with that little bit out of the way, we're going to think of dendritic spines as excitatory synapses, because they usually are. The dendritic spine is going to have two parts. It's going to have the spine head, and that's where we have the postsynaptic density. And it's going to have the spine neck. And that's where we're going to connect the head to the dendritic shaft, and that's where we're going to find the spine apparatus, for example. So here's our dendrite. I don't know if that's showing up. Let me do this. So here's the shaft of the dendrite, so just dendrite. Here's our neck, and here's the head of the spine. So here's our dendritic spine right here. Some are going to be kind of mushroom shaped, some will be stubby, some will be thin, some will be Philippodia-like. We'll see those in the next slide. The head is where we have the postsynaptic density. So these look electron dense, that's why we call them that, and that's because they're filled with proteins. We're going to find neurotransmitter receptors and scaffolding proteins in the spine head. We're also going to see a whole bunch of actin, but we won't see microtubules. So look at that colored picture on top. Red is showing you microtubules, that's filling the shaft, but in the head we find all that green actin. Actin is a much more dynamic cytoskeletal filament. And so this should tell you that spines can grow and shrink, and that's true. So we find a bunch of actin, we find neurotransmitter receptors, they're scaffolding proteins, and we find spine apparatus and smooth endoplasmic reticulum. The smooth ER and the spine apparatus are going to buffer calcium. And calcium needs to be buffered because it's a secondary messenger. So if we get calcium influx here, because this synapse was active, we want to keep it here, because that calcium is going to affect how that synapse works in the future. We're not going to find mitochondria, and we're not going to find ribosomes. Not in the spine, we're going to find it down here. So we can still make ATP, we can still make proteins, but it's out here. We're not going to invest in this, because spines can grow and shrink. We may very well find this spine gone later on. And that's the beauty of dendritic spines. They can grow and they can be eliminated, but notice we haven't gotten rid of our dendrite. So all the other spines on it can remain. New ones can grow, and they won't really have much of an effect on their neighbors. This is still going to have the same effect on the cell body as it did before this one was here. So this makes the neuron a lot more flexible in its ability to increase and decrease the strength of its synapses. Now, in general, the bigger the spine head, the stronger the synapse, and that's because with the bigger spine head, you can fit a whole lot more neurotransmitter receptors. You just simply have more space. So for thin or stubby spines, not as big as a mushroom spine. And in a mushroom spine, you're going to expect greater postsynaptic currents. Again, your synaptic current is a function of the open probability, the conductance, and also the number of ion channels that you have there. So, pack in more neurotransmitter receptor, get a stronger postsynaptic current. And those postsynaptic currents are going to translate to much greater changes in membrane potential because dendritic spines have very high internal resistance. Distal dendrites certainly have high internal resistance, but because of that very thin neck, we are going to have much higher internal resistance in the dendritic spine. And so we're going to see larger postsynaptic potentials as a result. So just take a look at the bottom. The postsynaptic potential that's created in the dendritic spine, that's shown on the left there, it's kind of pink looking. The trace on top is showing you the spine, and the trace below is showing you the shaft. So in the spine, we're going to see a much greater potential than we would out there in the shaft. And that's because we just simply have greater internal resistance, and this is still true. It was true 20 minutes ago, it's still true, and it'll probably be true 20 minutes from now. Now let's compare that to whenever we initiate a potential out here. Now if we start this potential from a synapse on the shaft, that actually seems to move into the spine just fine. So we get fairly unidirectional flow of charge. Because the spine amplifies that flow, that current that comes in, we're going to see a consistent depolarization within the spine as we do in the shaft. But when we have depolarization in the spine, it's much greater than we see in the shaft. And that cartoon shows you that. So on the on the right, now we have our synaptic input on the dendritic shaft. And you'll notice the top and bottom have basically the same amplitudes there. That means if we initiate an action potential, so if you look on the top now, top right, when we have a back-propagating action potential, that's of course going to start in the axon, move to the cell body, move to the dendrite, then into the spine. We see consistent depolarization in the spine, shown in red, and the dendrites, shown in black or gray. On the other hand, when we're looking at synaptic currents, those tend to be much greater in the spines. So whenever we initiate that current and create a potential in the spine, it's going to be much greater than in the shaft. So they're little amplifiers because of their high internal resistance. Now the charge spread is going to work fairly well. So whenever we create charge here, we can get fairly consistent depolarization in our neighboring synapses. That's what these data are showing us. So if we look at the data on the left, what we're doing is uncaging glutamate. And that really is what it sounds like. So they have a little molecular cage, and I'm actually going to draw it like a cage. It's not shaped like this, and it doesn't have little bars on it. Well, it kind of does. And inside is glutamate. And the only thing that will open this cage is the appropriate key, and the key in that case is blue light. So when you flash a little bit of blue light onto the bath there, the cage falls apart, and we're left with free glutamate. So you can release glutamate with using light. So let's look on the left. This is just essentially a control experiment. We're going to look at two spots. Here you can see a dendritic shaft, and there's some spines coming off. And then there's an area of nothing. So the kind of red, pinkish uncaging and light, they're flashing glutamate onto a spine, and they're looking at the spine. The yellow orangey one over here, they're flashing glutamate nowhere and looking nowhere. And when you do that, you get nothing. So look at the orange trace, it's basically a flat line. No calcium on top, no charge on top, and no calcium on the bottom. So if you're measuring nothing, you see nothing. That's great. We want to see that. What's a little more informative is looking at the kind of red orangish trace there where we're actually on the dendrite. When you flash glutamate on it, it depolarizes. Great, that's what we expect. And we get an influx of calcium. Cool, we'd also expect that too. Now let's look at the data on the right. We're looking at 3 different synapses. When we uncage glutamate on that green spot right there, so next to one of the dendritic spines, notice that they all depolarize. So that charge that comes in still spreads passively, but only the green one has calcium flux. That's down on the bottom. Largely this is going to be because of the spine apparatus. that spine apparatus is going to pick up the calcium, so that only in the spines where we actually have glutamate input do we see an increase in calcium. And only those are going to be strengthened, and only those are going to be modified. So this makes our learning a little more specific. Now that spread of the charge means that if these also receive glutamate at the same time, well, they would get calcium too, and they would be strengthened. A lot more on this in Lecture 10. What we need to take away from this right now is that dendritic spines amplify postsynaptic potentials because of their high internal resistance. And although they allow the spread of charge to go to neighboring areas, and they accept charge from the dendrites, they don't let calcium leave. They create these little, essentially, pockets of calcium where they get an increase but no one else does, unless they also get a neurotransmitter. They get the charge but not the calcium. This will be fairly important when we talk about learning. Speaking of learning, I think we've probably learned enough so far, so we're going to call it quits here. If you have any questions for me, fill out the questions box. See you in class.